Welcome to our GIT histology lecture. Today we are going to focus on the small intestine histology as well as the large intestine histology. So, the small intestines, the their main function is for absorption. So, you're going to see some structures in the mucosa of the small intestines that facilitates the absorption function. Is it? So the small intestines are going to be divided into three. You have your duodenum, your jejunum, and your ileum. Is it? Right. So the function: terminal food digestion, nutrient absorption, as well as endocrine secretion. Very important. So let's take a look at the small intestine. Histologically, the small intestines they have permanent folds that are called plicae secularis. Plicae secularis, these are permanent folds of the mucosa and the submucosa. And these they increase the surface area of the small intestines by threefold. By threefold. You understand? So the core of the plicae secularis is going to be made by the submucosa. Very important. Submucosa, as we're going to see later. And from the plicae secularis, you're going to have intestinal villi. The villi is a core of the lamina propria with a central lymphatic called the lactea, and it's going to be lined by epithelia of simple columnar epithelia. And this is going to increase the, the surface area by tenfold. Tenfold. Right. So take note of this. As we move from the duodenum to the ileum, the villi decrease its height. You understand? So in the duodenum, you have the villi that is about 1.5 millimeters, but in the ileum, you now have a villi of 0.5 millimeters. Why? Because most of the absorption takes place in the duodenum and the jejunum. So that's where we have long, numerous villi. The core of the villus is going to be made by capillary loops, blind ending lymphatic channels, the lactea, and some smooth muscle cells rich in lymphoid cells. Take note of this. The plicae secularis and the villi, they are permanent structures of the small intestines. The villi epithelia is continuous with the invagination called the crypts of Lieberkühn. So between a villi, there's gonna be a space that looks like a cleft. Those in that space, we are going to have the crypts of Lieberkühn. The what? The crypts of Lieberkühn. So they are short tubular glands with an epithelia which is continuous with the villi. Which epithelia? Simple columnar epithelia. Right. The epithelia of these crypts is pluripotent. So which means it has stem cells that have the capacity to regenerate the epithelia of the small intestines. So in these crypts of Lieberkühn, you are going to have the following cells. You are going to have the goblet cells, which are responsible for mucus secretion. You are going to have regenerative cells, as stem cells, which are pluripotent cells for regeneration of the epithelia. And then you're going to have these Paneth cells. So let's look a bit on the Paneth cells. So Paneth cells are located at the basal portion of the crypts of Lieberkühn, and these they secrete what you call defensin, right? So these are substances that protects the small intestines. The substance that includes lysosomes, phospholipase A, zymogens, zinc. These are substances produced by the Paneth cells. Take note of this. The apical portion of the Paneth cell is eosinophilic, but the basal portion of the Paneth cell is basophilic. Right. And then another important cells, we have the M cells that are the microfold cells. The microfold cells, they have presence of basal membrane pockets containing many intraepithelial lymphocytes, and they are going to communicate with pair patches which are found in the submucosa. Right. So those are the crypts of Lieberkühn. You need to understand them. Right. So right in the small intestines, we have absorptive cells that are your enterocytes, your tall columnar cells. They have a brush border appearance of microvilli to increase the surface area. So they have a brush border, and the brush border is going to be covered by a glycocalyx substance, which also facilitate absorption. These microvilli, they are outgrowth of the epithelium, and they increase the surface area by 20-fold. 20-fold. What is found on the core, in the core of the microvilli, these are actin filament. So, how many folds? We spoke of the plica secularis, and we said the plica secularis, these are permanent fold with the core of a submucosa. And then we spoke of the villus. Villi said also these are permanent folds that are made by the core of the lamina propria with the lymphatic lacteus. And then we spoke of now the microvilli, which are also permanent folds that are made by the core of actin filament. Very important. You need to understand those things. Right. So the small intestines have a total surface area of 200 meters squared, which is similar as a tennis court. So the surface area of the small intestines is similar as the area of the tennis court. Right? Good. So function is the terminal digestion and absorption of water and nutrients, re-estify fatty acids into triglycerides, right? Form chylomicrons, transport absorbed nutrients into the lamina propria and enter the blood vessels. So we also have these cells, like I said, the goblet cells, which are also found in the crypts of Lieberkühn, right? They will be interspersed between the enterocytes, and they increase as we go down. So goblet cells, as you move from the intestines, small intestines, the large intestines, the rectum, they will be increasing their number, right? So their function is to protect and to lubricate the small intestinal lining. We also have the Paneth cells, which you say that mostly found at the base, and they are eosinophilic at the apical and the basophilic at the base. Regenerative cells, these are pluripotent stem cells that regenerate the small intestines, right? And they have few organelles, many free ribosomes, and they have a high rate of cell division. Take note of this. Take note that the regeneration takes uh, after every 5 to 7 days, right? That's where there's a regeneration of the small intestine mucosa. We spoke of the M cells. We said these are specialized epithelial cells that overlie lymphoid follicles of pair patches. So they have numerous basal membrane invagination, which are called pockets, right? That present antigens to the antigen presenting cells, right? So they lie on a discontinuous basal membrane which facilitates transit between the lamina propria and the M cell via those pockets, right? So we also have other line of defense. For example, we have plasma cells that will be secreting immunoglobulin A class, and we have the mucosal lining, the lysosome, which are produced by the Paneth cells, and then we have the main protective layer, which is called the gout, right? The gastro-associated lymphatic uh, uh, system, right? That will be secreting immunoglobulin from the plasma cells. There's macrophages and lymphocytes, both in the mucosa and the submucosa. And then we have those cells that are responsible for producing hormones. Different hormones are produced by the small intestines and aside, for example, coli, coli, for example, these hormones are secreted by these enteroendocrine cells, right? So they make up 1% of the cells covering the villi and the intervilli space. That is your crypt of Lieberkühn. Let's take a look at the submucosa. So the submucosa has dense, irregular, fibrous connective tissue, which is rich in large vascular and lymphatic vessels, and it houses the Meissner plexus, remember, or the submucosal plexus. And in the duodenum, we have these Brunner's glands that are unique to the duodenum. These Brunner's glands, they can also be found in the mucosa and the submucosa of the duodenum. They are branched tubular-alveolar glands which penetrate the muscularis mucosa, pierce the base of the crypts to empty the duodenal lumen. So their function, they secrete an alkaline fluid, right, that is going to neutralize the chyme that will be coming from the stomach. And then and our muscularis externa has two layers as usual: inner circular, outer longitudinal smooth muscle, and then between the inner and the outer we have the mesenteric or the Albrecht plexus located between the layers, right? And then see the small intestines, they are covered by what we call mesenteric proper, which is a mesentery. They are going to be lined by serosa, except the second, third, and fourth part of the duodenum. Only the first part of the duodenum is referred to as intraperitoneal. Right. Let's also take a look at the lymphatic and the vascular supply. So the lymphatic and the vascular supply, they are found in the submucosa as well as the lamina propria. We have the capillary loops in the villi, which drain the blood vessels, which drain into the blood vessels. Now let's take a look at some differences between the duodenum, the jejunum, and the ileum. So the duodenum is the shortest segment. It receives bile, remember, from the bile duct at the major duodenal papilla with some pancreatic juice, right? And he has broader and taller numerous villi. And we also say that the Brannus gland in the submucosa, the prime. So then the jejunum has villi, narrow and shorter. So the villi, as you move from the duodenum to the ileum, it decreases in number and in size. But as for the goblet cells, they decrease in number as you move from the duodenum to the ileum, even unto the large intestines. Right. Let's take a look at the ileum. So the villi in the ileum are the narrowest and shortest, right? And what is distinct in the ileum? You find these permanent clusters of lymphoid nodules in the antimesenteric side called pious patches. They are just unique to the ileum. So we are now getting into the large intestinal tract, the large intestines. So the large intestines, they compose of the cecum, colon, that you're ascending, transverse, and descending, and sigmoid. And we're going to talk about the rectum, the anus, the upper half of the anal canal. You understand? Right. Their main function is to absorb water and to concentrate fecal matter. So let's take a look at some features of the large intestine. Now, first of all, the diameter of the large intestine is about 6 to 7 centimeters with walls that are packed into hyastra. So you're gonna see some dilatations as I'm gonna show you some pictures that are called the hyastra, they are unique to the large intestine. We have no villi in the large intestines, but we have microvilli. We have the crypts of Lieberkühn that I have similar composition to those of the small intestines, except there is absence of Paneth cells. The number of goblet cells increases as you move from the cecum to the sigmoid, right? So we have abundant goblet cells in the large intestine, right? So this is your large intestine, small intestines. You can actually see these hyastra, these dilatations, right? And the epithelial lining is going to be simple columnar epithelium, and it's going to be interspersed by numerous goblet cells. So you can actually see numerous goblet cells here, right? And we also have these crypts of Lieberkühn, but with absence of Paneth cells. And this is going to be your lamina propria, your submucosa. And take note, there is abundance of lymphoid nodule. Remember, the large intestine has bacteria, if you remember, columnar bacteria. So we need these lymphocytes, lymphoid nodules, to act as a defense mechanism for these colonal bacteria. Let's take a look at the lamina propria. So like I said, it is rich in lymphoid nodules, right, due to the abundant bacterial population of the large intestine. As for the muscularis mucosa and the submucosa of the colon, it is just the same as that of the small intestine. But the muscularis externa is very unique. It is the outer longitudinal layer that is very unique, right? This outer longitudinal layer, it is gathered into 3 narrow ribbons of muscle fascicles called Taenia coli. So you're going to see these Taenia coli that are going to be found on the muscularis externa. 3 bands of smooth muscle. So you can actually appreciate, right, that this is your submucosa, your muscularis mucosa here, your mucosa here, right? And you can actually see some pits, your crypts of Lieberkühn, and simple columnar epithelia interspersed with goblet cells, right? And this is your submucosa. This is going to be your inner circular layer. Your outer circular layer arranged into 3 Taenia coli, and between you can actually appreciate the albec or the mesenteric plexus. Right? This is another histological slide. This is your mucosa. You can actually appreciate your muscularis mucosa here. Your muscularis mucosa right here. Your submucosa. This is going to be your inner circular layer, and you can actually see this bundle, which is called Taenia coli, unique to the colon. Another histological slide here. These are the crypts of Lieberkühn, which have absent Paneth cells, and you can see numerous goblet cells. Interspersed with the simple columnar epithelia that is absorptive, with the microvilli but absent of villi. Remember, this is your muscularis mucosa, very prominent. This is going to be your submucosa, where you're going to have large veins. This is a vein, this is an artery. Yeah. So you can actually appreciate this is a goblet cell right here, another goblet cell right here, another goblet cell right here, another goblet cell right here, and these are your colonocytes. These are your colonocytes. Colonocytes. You can see this is a columnar, oval-shaped nucleus, basally placed. You can actually see, right? Right. So another thing that is important and unique to the large intestine is its serosa. The ascending aorta and the ascending colon and the descending colon, they have adventitia and serosa. But as for the transverse colon, we have the serosa because it has the transverse mesocolon, right? Which is the mesentery. So the serosa has these appendix epiploids. These are fat-filled pouches, right? Let me see if they are clear here. Or not that clear, but we're gonna see it in the presentation where we are gonna do all the histological slides of the GIT. Now let's take a look at an anal canal. An anal canal is about 4 centimeters long. It's the terminal part of the digestive tract, right? And its mucosa has folds, longitudinal folds, that are called anal columns. Each column contains terminal branch of the superior rectal artery and vein. Right. So if you want to gauge the anal column, you need to divide the anal column into 2, the upper half and the lower half. And if you understand the embryological origin of the upper half and the lower half, it's going to be easy. The upper half comes from the ectoderm, the lower half comes from the ectoderm. So the upper half is going to be a part of the Hintergut. So therefore, it's going to be supplied by the inferior mesenteric artery via the superior rectal artery and vein. While the lower half is going to come from the ectoderm. Since it comes from the ectoderm, it's going to be supplied by the inferior rectal artery, which is a branch of the internal pudendal artery. Very important. So there's going to be a line between the upper half and the lower half because there's also transition of epithelia. In the upper half, you have simple columnar epithelia. In the lower half, you have the stratified squamous epithelia. So there's a line that's called a pectinate line that is going to divide those two parts, right? So you can actually see these are the columns, the anal columns that we were talking about. This is the pectinate line or the transitional line, where this portion, this upper half, is going to be simple columnar. This lower half is going to be stratified squamous epithelia. This is supplied by the superior rectal artery and vein. This is supplied by the inferior rectal artery and vein, right? This is associated with internal hemorrhoids. This is associated with external painful hemorrhoids, right? So the crypts disappear below the pectinal line. So below the pectinal line, we no longer have these crypts, right? But we now have the stratified squamous epithelium. And the muscularis externa gradually becomes thicker and forms involuntary internal anal sphincter formed by the inner circular layer, right? So you now actually see the demarcation. So this is going to be your pectinal line, that is your transition zone. This is your upper half, your lower half. You can actually see this was a simple columnar epithelia, right? And now we have stratified squamous epithelia, keratinized stratified squamous epithelia in the lower half, right? Another portion of the GIT which is very important is the appendix. So appendix is just a diverticulum that is found in the retrocolic from the retrocolic space, which is a small blind ending diverticulum from the cecum. Its important features: it is a small, narrow, irregular lumen, thickening of the wall, right, due to accumulation of lymphoid tissue in the lamina propria and mucosa. So in the appendix, what you're going to find is the lymphoid aggregation, the mount. There's going to be absence of intestinal villi and some few crypts of Lieberkühn. So the muscularis externa is thinner than in the remainder of the large intestines, and the outer longitudinal layer does not aggregate into Taenia coli. So the Taenia coli, they start at the base of the appendix, right, going upward. But right in the appendix, we have our longitudinal outer layer not branched into, not in the bundles of Taenia coli. So this is a cross section of a histological slide of an appendix. You can actually appreciate this is your muscularis externa, this is your submucosa, right? And this is your mucosa. And you can actually see aggregation of lymphoid nodules. You're going to see this germinal center right here, and your periphery of T cells in the germinal center right here, another lymphoid node here, another lymphoid node here, another lymphoid node here. So there's aggregation. And some few crypts of Lieberkühn with absence of villi and microvilli. Another one here, you can see crypts of Lieberkühn here. This is a lymphoid nodule. This is a muscularis mucosa, right? Right, so that's everything about the GIT histology. Thank you so much. See you next time.